Hello and welcome once again to AJP-Heart and Circ Table of Contents video highlights from the editor's desk for September 15th, 2014. Boy, do we have some great science for you this month. Here's a sample. Domeyer et al. Investigated an issue of functional remodeling in the aged heart. They examined dysfunction of the ryanodine receptor, calcium release, and diastolic dysfunction by using the ryanodine receptor inhibitor dantrolene. They found that dantrolene prevented spontaneous calcium sparks in aged hearts but not in young. The authors speculate that dantrolene may be an antiarrhythmic in aged hearts. Howe et al. Compared the effects of adipose-derived regenerative cells with bone marrow mononuclear cell transplantation in a rabbit model of hind limb ischemia. The data show that adipose-derived regenerative cells incorporated into the endothelial vasculature in vitro and in vivo. Both adipose-derived regenerative cell conditioned media and bone marrow mononuclear cell conditioned media enhanced the migratory ability and interrupted the process of apoptosis in human umbilical vein endothelial cells. The authors conclude that adipose-derived regenerative cell transplantation harvested similar effects of neovascularization augmentation compared with bone marrow mononuclear cells in experimental rabbit model of hind limb ischemia. Ho et al. Assessed the effects of graded cholesterol depletion and overexpression or deletion of caveolin-3 on cardiac function following ischemia reperfusion injury in mice. The data reveal membrane cholesterol dependence of normoxic myocardial and coronary function, ischemia reperfusion tolerance, and opioid receptor-mediated cardioprotection. In contrast, baseline function appears insensitive to caveolin-3, while cardiac ischemia reperfusion tolerance parallels caveolin-3 expression. Finally, Minnick and colleagues tested the hypothesis that during severe hemorrhage, cardiopulmonary chemoreflex control of sympathetic output is attenuated by adenosine, naturally released into the nucleus of the tractus solitarius. The reflex response to right atrial serotonin on sympathoinhibition was significantly attenuated during severe hemorrhage despite reciprocal changes in the baseline activity levels, and this attenuation was removed by bilateral blockade of adenosine receptors in the caudal NTS. These data confirm the importance of adenosine released in the NTS on cardiopulmonary reflexes. Once again, I thank you for listening and hope to see you in 2 weeks. And don't forget to send us your best work now.